Well, thank you for nominating me to present this work, also the recognition. Today I'm going to talk about, and hopefully the last steps of this project that is a tight collaboration between us and Germany, Dr. Stefan Pfister's group, and CHOP. So a very brief introduction. I'd just like to point out 3 recent papers came out in 2020 studying the efficacy and toxicity of immune checkpoint inhibitors as monotherapy in basket pediatric cancers. The NIVO trial reported partial responders in 4 cases of lymphoma and stable disease in about 11 of sarcomas and 5 neuroblastomas. The iMatrix trial run by Roche is another collaboration we have, and we're studying the data behind it, but they also reported 4 partial responders, 3 lymphomas, and 1 rapid tumor originating from thorax, also 11 stable disease, mostly sarcomas and neuroblastomas. And the keynote 51 study of pembrolizumab from Merck was the only trial included pediatric melanomas and also PD-L1 positive only cancers. And there they show complete or partial response in lymphomas, but also in about 8 cases of solid tumors, including a few CNS tumors, ganglioglioma, and a few others. But they also concluded that PD-L1 is not a sufficient biomarker to identify potential responders. So clearly a need for potential biomarkers of immune response in children that hopefully can help clinical trial designs, but also the precision oncology programs we have in different countries. So really the objective of our study was to really starting by asking how the overall layout of immune microenvironment looks like in a large number of pediatric cancer samples, and hopefully to learn more about them and inform the prospective cases. And so to do this, we compiled an RNA-seq-based data set of about over 900 cases, mostly CNS tumors, but also neuroblastomas. We focused on initial resections, primary tumors. We used the Kids First Data Resource Center, the harmonized data from CBTN, target anti-CGA, also included data from previously reported from ICGC and our collaborators from Germany. We primarily used Cabalica as a platform to do our analysis, also share the progress, results, and codes with our team and our collaborators. And again, using the resources from Cabalica, we included about 2,400 cases of adult cancers for TCGA consortium as a comparator to our study. So the ICGC data was extensively studied before, but CBTN was rather new to us at the time, CBTN or CBTTC back then. So out of over 1,000 cases from CBTTC, made available to us at the time, we removed about 34 that were annotated cell lines or originating from other tissues. About 8 or 3 initial CNS tumors were initial CNS tumors annotated as samples from initial resection, of which 77 were CNS tumors. So out of these, we further looked into the comparable purity and transcriptional profile and came up with a rather clean data set of 638, pooled it with ICGC data to perform our analysis. So how did we do the very last step of cleanup? We did this through unsupervised clustering. We identified tumor type clusters and see some samples are transcriptionally very different within the same cancer type. So this one Ewing sarcoma of cranial Ewing sarcoma or low-grade glioma clustered with rather clean medulloblastoma subtype. So we removed those and we redid analysis, and you see now we have a transcriptionally distinct tumor types that we pooled with the curated ICGC data to do our analysis. Okay, so we used estimate algorithm to look at overall immune infiltration and looked at the correlation with the overall mutation burden. Just to orient you, the top plot is the percentage immune reads in each sample. The bottom plot shows the mutation number per megabase. In cases ordered same as the plot above, just to so you see the associations between the TMB and immune infiltrate within each case. We included a number of RNA-seq data from medulloblastoma PDX models that are virtually no cells in there, served as a negative control just to make sure that what we see is meaningful. 2 embryonal tumors, medullos and ETMRs, known to be hybrid pure. You see on the far left has, if any, minimum immune infiltrate compared to other pediatric cancers and also adult cancers. And through this landscape, we also noticed the HRT as sort of a interesting tumor entity, also published before, that had a wide range of immune infiltrate independent of TMB. Okay, so overall, immune infiltration baseline really seems to be independent of tumor microenvironment across pediatric cancers. And then we looked a little deeper into the type of immune cells within these samples using the RNA-seq data. And what you see here is the median infiltration of 9 immune cell types across pediatric cancers and adult cancers. This analysis, as you see now, is including only cancers with more than 5% immune reads as sort of detectable immune infiltration at this resolution of bulk RNA. Just point to a few observations, interest of time. The few cases of medullos have sufficient immune reads. We see some CD4 T cells, NK cells, likely group 3, group 4 medullos. Here, we recovered a lot of NK signal from neuroblastomas as known previously from other publications, mostly non-amplified neuroblastomas, but also in schwannomas. And also in craniopharyngiomas, we recovered a lot of neutrophils compared to other CNS tumors. Just an indication that might have a play role in immunosuppression in these cancers. And so just to showcase the value of really proper clinical data available alongside the data as is with CBTN and ICGC, we looked at the survival value of immune infiltration in these cases and find in a multivariable model. Again, in cases with detectable immune infiltration, in both neuroblastomas and high-grade gliomas, a high NK in the context of low macrophages are prognostic. And at this point, we are looking more into mechanisms of this type of observations. And just to show another little layout of our study, using RNA-seq, we looked at the infiltrating T and B cell repertoire. We developed a robust estimate of T cell diversity using the bulk RNA. It could reliably estimate the immune diversity in about 330 samples. And there's a high correlation between the TCR reads recovered in RNA-seq to the overall T cell diversity. But we were able to look at the outliers at both ends. In other words, in any range of TCR reads, there are overly diverse samples with significantly low diversity that are annotated here. Most of them are extracranial neuroblastomas, but also cranial, rhabdoid tumors, few neurofibromas. And the circle plots here just to show how the underlying repertoire shape would look like, and you just see that you see here much more polyclonal infiltrate compared to here. Is it a indication of antigen and specific expansion, or can lead to a discovery of new antigens in this type of tumors? Something that we are looking But really, this is work in progress. We now have a public project on Kvaliko where we store all our process data and are working to share all analysis and figures. It's just a short description we have already on the public project. You can access it here. Hopefully, we plan to use the data cruncher feature on Kvaliko to help us with the figures and with the reproducibility, sharing codes as well, and also sharing the apps. This is just a app that we designed to do the immune repertoire analysis. By sharing these, hopefully it reduces the time and effort for the community, but also saves some dollars. People don't have to pay again and again to do the same jobs and analysis. Really, the hope at the end is to have this as a public workspace for pediatric immunogenomics and expand it to other cancer types as the data becomes available. I hope I am not over time that much, but to summarize what I've shown you, we find the highly variable microenvironment in pediatric nervous system tumors highlights the value of personalized profiling, particularly maybe as an add-on to molecular tumor boards we already have with different precision oncology programs, Profile in Canada, Zero Childhood in Australia, or Inform in Germany. And also really shows the importance of appropriate sample data / clinical data collection. To our experience, CBTN has been a great resource. I think three angles that has been really helpful at least from a trainee perspective was the GIFT's first data resource center where we could actually browse what data is out there, what the progress of what the processing would look like, Kvaliko as a computing platform, but also the repository query system where you can actually look up and very quickly see what samples they are and what kind of clinical data are associated with these samples. With this sort of a setup, we think we can ask a lot of interesting questions. So one of the questions we are also interested in looking into in future is to look at the effects of prior therapy on tumor microenvironment and immune response, but also looking at the evolution of tumor microenvironment in CNS tumors over time. With that, I hope I am on time. But I'd like to thank my mentor, Dr. Trevor Pugh, and his lab. This work was nearly impossible without the great collaboration between us, Heidelberg and Dr. Fischer's group and Natalie Yeager, but also CBTN and Kids First Data Resource Center. I've been very lucky that the release of Kvaliko was very well aligned with the first year of my postdoc. So to be honest, I learned a lot of cloud computing with Kvaliko. I also learned a lot about the clinical data collection, curation with CBTC. With that, I'd like to thank Adam Resnick for providing this opportunity, plus everyone that I have contacted over the years. As soon as I faced the little issue, Pichai and his team for CBio, Yonkon and Michele for cloud problems, the data processing, and Allison, Jennifer, and Bailey for any questions on clinical data. With that, I'll stop and take questions.
Thank you so much, Arash. That was a really beautiful representation of the interoperability of these data sets when they're properly harmonized and brought to the public. Really empowers some pretty meaningful research that in your case really has significant translational impact. Your timing, by the way, was perfect. Thank you very much. And we probably have time for a question if anyone wanted to pose one in the chat or simply unmute themselves and speak.
Arash, I've been a huge fan of your engagement, of this resource. As you mentioned, one of our main goals is to, quote unquote, brainwash and ensure young trainees commit the rest of their lives towards pediatric brain tumor research. And we're going to count you as one such successful acquisition into that landscape. But maybe to take a more critical look, maybe you can comment, what are some of the challenges or opportunities that we should be focused on improving on. You've been extremely complimentary, but I'd love to hear also, what are things that, can enhance your support, of a young trainee who is engaging these efforts in a meaningful way?
I think, like I said, I learned a lot of cloud computing with Kvatika, and since then I have tried and learned other platforms out there. But so far, I would say from a biologist standpoint, just for people who don't know me, my background is cell biology. When I came to Trevor's lab, I didn't know anything about bioinformatics. I would say the documentation, the support of Kvatika is really outstanding. The documentation, as I'm pretty sure if Yankon or Michele are here, I've said multiple times, the documentation is just well suited for anyone with any level of expertise. If you're a computer scientist, documentation is out there for you. If you're a biologist and want to, just do the app with drag and drop, documentation is there for you. I think that's really great in that sense. With regard to challenges, I think it has been much more streamlined now. When we started, it was just us, I guess, and there was a lot of, I guess, communication needed. But at this point now, I'm seeing that Kids First, the portal, and Kvatika really are complementary together, work there. I can't really think of anything. I mean, the platform really, there are, at times that there is a link broken between the Kids First and Kvatika that would, cause a little bit of problem. When you want to spin off the cluster, it takes forever. It's like all these little issues with any structure, I would say. But I think from a biology standpoint, I think people should not be scared of cloud computing, I guess, and 7 Bridges and Kvatika would be really a good way to start. Now, whether or not that's financially feasible, that's something that I can't comment on. But if the dollars are there, it's perfect, really.
Perfect. And I promise I didn't pay Arash to say any of those things. But I am glad that you ended at that point, which is, and this is something we're trying to even address in the context of NIH. And it's one of the things that we've been very privileged through the Kids First program is to support the storage, compute, analysis. These things still cost money to do these things in ways that there is no a priori model for that. And so it's still, in fact, the Kids First program piloted these efforts with us on how do you provide resources to investigators to do these large scale compute and analytics to engage them. And I think it's still an evolving narrative, and certainly several foundations and philanthropists have been supportive of this process as well.
I think that's more important, that anyone that uses the platform should be able to share their process data. I don't think the analytics would take that much money from my own experience, but the jobs that I ran OptiType for over 1,000 samples, and another investigator should not really have to do that, and can use my process data. So
yeah, perfect. And this relates very closely to also the work that Jacqueline and the Data Lab